Welcome back to the Science Channel podcast, where we discuss the coolest scientific papers and preprints of our day.
Great to be back.
If you're finding these deep dives into science fascinating, please do take a moment to like the video and subscribe. It really helps us grow.
We appreciate your support. Seriously.
Today, we're diving into a paper that has really made me rethink something fundamental. Mitochondria.
The good old powerhouses of the cell.
Exactly. But we all learned they're the cell's powerhouses, and it turns out that's a massive oversimplification.
It really is. There's so much more to them.
Yeah, this recent paper, A Quantitative Approach to Mapping Mitochondrial Specialization and Plasticity. It reveals a surprising level of diversity among these tiny organelles.
It's a fascinating piece of work. It introduces a new computational method called mitotyping.
Mitotyping, okay.
Yeah, it's basically a way to get a detailed molecular profile, like a fingerprint, of different types of mitochondria. So our goal today is to explore how this technique illuminates the specialization of mitochondria across various tissues in the body.
Okay.
And even how they can change within cells over time. It's pretty dynamic.
All right, let's unpack this then. The paper highlights that just like the different organs in our bodies have specific jobs.
Right, like muscles for movement, liver for processing.
Really for thinking, yeah. Mitochondria also develop specialized roles. They call them mitotypes.
Exactly, mitotypes. And what's fascinating here, really, is that all these varied mitochondria, they actually originate from the same source.
Which is?
The mitochondria that are already present in the egg cell.
Ah, okay, from the very beginning.
Right. So this initial flexibility, this plasticity, allows them to adapt in really remarkable ways to the specific needs of different cells and tissues later on.
So this specialization isn't always about something going wrong, like dysfunction.
No, not at all. Often it's the cell's way of optimizing its function. It's an adaptation to its specific job or environment.
Okay, so how do scientists actually, identify and measure these mitotypes? That's where this new mitotyping method comes in.
Precisely. Mitotyping is a quantitative approach. It puts numbers on the degree of molecular specialization among different mitochondrial types.
Okay.
So the researchers developed what they call MitoPathway scores.
MitoPathway scores. Got it.
These scores are calculated by looking at the average activity levels. Well, the expression levels of genes within specific mitochondrial pathways.
And they know these pathways from?
They use a comprehensive database, Mitocarta 3.0. Think of it as like the encyclopedia of all known mitochondrial genes and proteins. It's very thorough.
Okay, so you look at which genes are active, group them by pathway using Mitocarta, and that gives you a score.
Exactly. You average the expression within each defined pathway.
So by looking at which pathways are most active based on these scores, we can get a snapshot of what a mitochondrion is really geared up to do in a specific situation.
That's the idea. Because Mitocarta 3.0 is so comprehensive, these MitoPathway scores give us a really broad picture of what's happening inside.
Right.
And then by comparing these scores across different samples, like different tissues, we can see which pathways are, prioritized in each context.
Which leads to a particularly interesting finding, I thought. The researchers used this mitotyping to analyze mitochondrial protein levels in, 14 mouse tissues?
Yeah, and gene expression in a remarkable 55 human tissues. A huge data set.
Wow. And they discovered they could actually distinguish the different tissues just by looking at their mitochondrial proteins and genes.
That really blew me away, too. It underscores just how distinct the mitochondrial makeup is in different parts of the body. It's not just a generic organelle.
So what were the main patterns they saw?
Well, their analysis revealed two primary ways, or axes, that mitochondria specialize across these tissues.
Okay, axis one.
Axis one is basically contrasting tissues that are primarily anabolic, like the liver, focused on building molecules, with those that are catabolic, like the brain, focused on breaking them down for energy.
Build versus burn, essentially.
Or much, yeah.
And the second axis?
The second key difference separates contractile muscles, think heart and skeletal muscle, from most other tissues.
Okay, so muscles have their own thing going on, too.
Definitely. They observe distinct clusters of mitotypes for the brain, for these anabolic tissues, for the contractile muscles, and then a sort of mixed, more diverse group for the remaining tissues.
So the big takeaway here is that mitochondrial specialization across tissues seems to be driven by sort of two fundamental needs.
That's what it looks like. First, whether the tissue is mainly building things up or breaking them down.
Anabolic versus catabolic.
Right. And second, whether it's involved in physical contraction.
Makes sense. To really illustrate how different these mitotypes can be, the paper zooms in on an example, right? Liver versus brain.
Exactly. They looked at a process called fatty acid oxidation, or FAO.
Which is how cells burn fats for energy.
Right. And what's striking here is the sheer scale of the difference in FAO activity.
How big a difference?
The FAO scores were found to be several times higher in the liver compared to the brain.
Wow.
Even within the same individual, which, makes perfect sense when you think about it.
Yeah, the liver's role in processing fats.
Exactly, compared to the brain, which mainly runs on glucose.
So the mitochondria reflect that fuel preference.
Very clearly. But they didn't stop there. They dug deeper.
Okay.
They investigated the relationship between this FAO pathway and the electron transport chain, the ETC.
Which is the main ATP generating machinery, the actual powerhouse part.
Right. And they found that liver mitochondria have higher levels of a specific enzyme, ETFDH.
ETFDH. Okay, what does that do?
Think of it as a specific loader. It helps transfer electrons, essentially fuel, from fat breakdown into the ETC.
Gotcha.
And liver mitochondria have more of this ETFDH relative to one of the main ETC components, complex I, compared to brain mitochondria.
So it's like the loading dock for fat fuel is bigger in the liver's mitochondria.
Sort of, yeah. It suggests a very fine-tuned, tissue-specific way of managing electron flow within the mitochondria, depending on the main fuel being used.
Interesting.
And they also noted more of complex II and other enzymes that use a specific cofactor, FAD, in liver mitochondria, which further supports these adaptations for handling different fuel sources.
Now, the researchers realized that just looking at these overall MitoPathway scores might be a bit, potentially misleading, right?
Yeah, there were some limitations. For instance, a tissue that just happens to have loads of mitochondria might show high scores across the board, even if no single pathway is particularly prioritized.
Right, it just has more of everything.
Exactly. So they developed a new measure called mitochondrial pathway prioritization scores, or MitoPPS.
MitoPPS. Okay, how does that help?
This was a really insightful step, I think. While MitoPathway scores tell us the overall activity level, MitoPPS is like zooming in on what's most important for that specific mitochondrion relative to everything else it's doing, and regardless of how many mitochondria are in the cell overall.
So it's a relative measure.
Yes.
Yeah.
It essentially tells you how much a particular pathway is prioritized in a given sample compared to all other pathways in that sample, and also relative to other samples. It normalizes things.
Okay, that sounds much more nuanced.
Yeah.
And what were some of the key insights they gained using this MitoPPS metric?
Well, some really interesting findings popped out. One was that brain tissues showed the highest prioritization of oxidative phosphorylation, OxPhos, the main energy production process.
Even though they don't use fats as much.
Right, but what they do prioritize highly is squeezing every bit of energy out via OxPhos. Surprisingly, immune cells showed the lowest OxPhos prioritization using this measure.
What else?
They also found, maybe less surprisingly, that the liver and some digestive tissues highly prioritize heme synthesis and processing.
Which makes sense, liver's role in blood and detoxification.
Exactly, and contractile muscles like heart and skeletal muscle, along with the pancreas, showed a high prioritization of creatine metabolism.
Which is key for quick energy bursts in muscle.
Precisely. And another interesting one, brain tissues, except for one specific area, the choroid plexus,
Okay.
and the liver both prioritized folate and 1-carbon metabolism, which are involved in building blocks for DNA and other molecules.
So different tissues clearly prioritize different sets of tasks for their mitochondria.
Absolutely. They even used a statistical technique, PCA, principal component analysis, on the MitoPPS data.
What did that show? Did it confirm the patterns?
Yeah, largely it did. The PCA results again highlighted how distinct the brain mitotype is.
Okay.
It also clearly separated out the immune tissues, the anabolic tissues, especially the liver stood out again, and the contractile muscle tissues based on their overall MitoPPS profiles.
So the statistics backed up the pathway level view.
Right. These different statistical components, the principal components, they reflected different aspects of pathway prioritization, like the balance between making energy versus maintaining the mitochondrial machinery itself, or prioritizing different metabolic routes.
Okay, so we've established big differences between tissues, but the paper also explored how mitochondria within the same cell type can change over time. They looked at aging, right?
Exactly. They shifted focus to aging in human fibroblasts. That's a type of connective tissue cell grown in the lab.
And they saw signs of aging in these cells.
Yeah, they observed the typical hallmarks of cellular aging over long-term culture. And they found that the mitochondrial aging in these cells seems to follow at least a two-phase pattern.
Not just a simple decline.
Apparently not. And individual donor cells showed slightly different aging trajectories, which is also interesting.
But were there common themes in how the mitochondria changed with age?
Yes, they did identify pathways that were consistently affected. For example, things related to mitochondrial quality control, like fission, which is splitting mitochondria, and mitophagy, which is clearing out damaged ones, those tended to increase.
Like the cell is trying harder to manage mitochondrial health.
Potentially, yeah. While pathways involved in maintaining the mitochondrial DNA and repairing it tended to decrease with age.
Okay. And did these aging fibroblast mitotypes end up resembling any of the tissue-specific mitotypes they'd mapped out?
This was intriguing. When they compared the aging fibroblast mitotypes to the different tissue types, they saw a trend.
Towards what?
Towards a more catabolic, sort of brain-like phenotype as the cells aged.
Really? So aging cells shift their mitochondrial priorities towards something resembling the brain's profile.
That's what the data suggested. A potential shift in mitochondrial priorities as cells get older, maybe reflecting changing metabolic demands or stresses.
Fascinating. Finally, the researchers looked at how these fibroblast mitotypes respond to, like, direct challenges, right?
Yeah.
Experimental conditions.
Yes. They wanted to see how plastic these mitotypes are in the short term. They treated the fibroblasts with various things, inhibitors of energy production, genetic mutations found in mitochondrial diseases, low oxygen, different nutrients.
And did the mitotypes change?
They did. For example, when they inhibited glycolysis, the sugar-burning pathway, using inhibitors like 2DG, or by forcing cells to use other fuels like galactose or BHB.
Ketone bodies, right?
Right. The fibroblasts showed a greater prioritization of OxPhos-related pathways, and their overall mitotype profile shifted somewhat towards that of the brain mitotype again.
So stressing their energy production pushed them towards that brain-like state, too.
It seems so. Now, it's worth noting, these changes in fibroblasts were generally smaller in magnitude than the huge differences they saw between distinct tissues.
Okay, so they're adaptable, but maybe not as adaptable as differentiating into a whole new tissue type.
Exactly. But it clearly showed that even within a single cell type, mitochondria are incredibly responsive and can recalibrate their molecular profiles based on the environment and cellular needs. Some pathways showed really significant changes even in those fibroblast experiments.
Okay, so to kind of wrap this up.
Yeah.
It's crystal clear that mitochondria are not just these generic powerhouses we learned about in basic bio.
Absolutely not. They are highly diverse, highly specialized organelles.
And their specialization, their mitotype, really depends on the tissue they're in, reflecting the unique metabolic job of that tissue.
Precisely. And this new mitotyping approach, particularly using those MitoPPS scores, gives us a powerful new quantitative way to study this diversity and also their flexibility, their plasticity.
Right. And it's fascinating that even within one cell type, like those fibroblasts, mitochondria can dynamically adjust their molecular profiles. They respond to aging. They respond to environmental cues.
Yep. Although, as we said, the scale of that change within a cell type seems generally smaller than the fundamental differences we observe between, say, a liver cell and a brain cell.
Still, it shows they're constantly tuning themselves.
Constantly adapting.
And the researchers have even made their mitotypeexplorer.org resource and the code available.
Yeah, it's all open access, a fantastic resource for the community if you want to explore the data yourself.
That's great. It really makes you wonder, doesn't it, how this dynamic nature of mitochondria might play into, well, everything. Normal physiology, aging, disease.
Oh, definitely. The implications are potentially huge.
What further insights might we gain by exploring these mitotypes in even more detail, maybe in different disease states or developmental stages? Definitely some food for thought there.
Indeed. Lots more to discover.
Well, that concludes our deep dive into the fascinating world of mitochondrial specialization and plasticity. Thanks for walking us through that.
My pleasure. It's exciting stuff.
Please don't forget to like the video and subscribe if you want more content like this.